So I want to take a moment to quickly just share our mission statement with you. There's some, the founders of KBI back in essentially 2004 for their contract services have a lot of passion about what they've done and how we can help patients, and we have a number of patient forums. So our mission is to accelerate the development of innovative discoveries into life-changing products to expand global access to medicine. So we like to believe that without some of the work we do, some of these medicines may never make it through the clinic. So we have a number of client partners that we really work with a lot of flexibility to help them through, whereas otherwise they may not. So that's kind of our mission. So KBI was founded on our foundation of analytics, of analytical characterization, formulation development, and drug substance and drug product stability. So that's what really got the company off the ground in 2004, and is still a core strength today. So that started in Durham, North Carolina, where we also have a mass spec core facility as part of that, and cell line development and GMP manufacturing and testing since 2010. In 2013, the process development labs needed more space, so we set up labs in RTP, North Carolina, which is where I'm based. And there we have mammalian, at this time, process and analytical development, some very high throughput, high powered development there. And Abhinav Shukla, who runs this organization, previously at Amgen and BMS, developing a lot of MAB platforms and non-MAB platforms. He brings a lot of focus on the late phase. So we've done quite a bit of process characterization and scale down process validation in the last couple of years. Our Boulder site we acquired in 2014. That was previously a Merck site, and with that acquisition we retained some really, really talented staff. The R&D group there has some really incredible scientists who are experts in fermentation expression, refolding with mass spec capabilities to monitor a product through refold. So some real horsepower there, and some ex-Amgen folks as well. So the manufacturing there is 1,500 liter stainless steel and associated recovery and downstream, and then a 300 liter single-use fermentation suite that's being built out now. Will be ready this fall, in which we also plan to have a 100 liter stainless steel fermenter for clinical products. The 1,500 liter scale is clinical and commercial. Also in Boulder, we have a particle characterization group that is really, truly world-class. Amber Fracken runs that group. They work a lot with the University of Boulder, Colorado. So for drug substance, particle characterization, drug product, a lot of horsepower there. And also a modeling and simulation group. We've got a core of 7 ex-Amgen staff that really have some impressive plant simulation modeling and mechanistic modeling capabilities. So in the Woodlands, Texas, as of February of this year, we acquired Opexa Therapeutics and some of their key staff. It's a site, they can do phase 1, phase 2 manufacture for cell therapy products in the Woodlands, Texas. And some really excellent scientists are there, and we also do cell-based assays out of that facility, as well as our Durham facility. Okay, so to quickly just run through some of our experience for you, I mentioned the analytical foundation. We call that AFS, Analytical Formulation and Stability. So our analytical work, we've covered over 220 molecules, of which over 100 antibodies and a number of other more complex proteins. For formulation development, I can truly say this is some of the best formulation development out there. Wayne Yount runs this group. He's here with us this week. We've done over 110 distinct molecules, and of those, 60 were antibody products, and of the total, 38 were lyophilized, and 30 were high concentration, up to 200 mg per mil. And then a number of these are late stage and commercial products, so it's well known for our formulation. Process and analytical development on the mammalian side, we've done over 80 molecules, including first in human, early phase rapid development, and high throughput late phase development. And then cell line development, we've actually been doing this for about 5 years at KBI, and we've done over 30 molecules, either using a client's own host system, the big pharma sending us their host and vectors, or using the Freedom CHO, but we also have a KBI DG44 based system. And then the mammalian manufacturer, we've done over 24 products now in our 3 by 2,000 liter facility in Durham, North Carolina, and I'll touch on our microbial experience in just a moment. So our mammalian development experience just gives you a feel for the type of molecules and type of programs. So, about half MABs, but I will say the light blue here, or the bright blue, the bispecifics we've done a lot more of. in particular over the last couple of years, and we have 8 or 10 biospecifics in development as we speak, our development or manufacture. And then a number of program types, anywhere from cell line development all the way through manufacture, the gene to GMP, or transferring in a more mature process and modifying that for facility fit. On the microbial side, in Boulder, we've done over clinical development for over 50 recombinant proteins, 10 of those to manufacture with lots of